This research article, titled Identification of Reference microRNAs in Skeletal Muscle of a Canine Model of Duchenne Muscular Dystrophy, published in Wellcome Open Research, explores the crucial topic of identifying stable reference microRNAs, or miRs, for use in studies of Duchenne muscular dystrophy, or DMD, using a canine model. DMD is a severe, ultimately fatal, muscle-wasting disease caused by mutations in the dystrophin gene. The study focuses on the D50MD dog, which is a valuable translational model because it closely mimics the disease progression seen in human DMD patients. The primary goal of this research is to establish a reliable set of reference miRs that can be used to accurately measure the expression of other miRs in muscle tissue. This is important because miRs are small RNA molecules that play a key role in regulating gene expression, and their levels are often altered in DMD. Accurate quantification of these changes requires a reliable baseline for comparison. The authors emphasize that identifying stable reference miRs is essential for ensuring the reliability of data in the study of DMD and in preclinical drug trials. They note that while circulating miRs in serum can be useful biomarkers for disease progression, measuring miRs directly in muscle tissue provides the advantage of assessing expression without the confounding factors of muscle mass and membrane integrity, also allowing for direct mechanistic insight into the roles of miRs within DMD-affected muscle. The introduction explains that microRNAs, which are approximately 22 nucleotides in length, are involved in gene expression regulation and are a potential source of biomarkers for neuromuscular disorders. Alterations in miR expression in affected muscles and circulation have been associated with DMD. DMD is caused by mutations in the dystrophin gene, which leads to a lack of functional dystrophin protein, a protein that is critical for normal muscle function. The lack of dystrophin results in repeated cycles of muscle damage and degeneration, eventually leading to muscle loss. The authors discuss dystromy-RS, which include miR1, miR133A, and miR206, noting that their expression is altered in dystrophic tissues. While dystromy-RS are elevated in the serum of DMD patients, their expression in muscle tissue is typically lower, unaltered, or mildly upregulated. The authors also discuss the role of miRs in muscle fibrosis, which is a prominent feature of DMD. Fibrosis is characterized by excess deposition of extracellular matrix proteins. Specific miRs such as miR21, miR29, miR199A5P and miR214-3P are associated with fibrosis in DMD muscle. The researchers emphasize the need for reliable biomarkers and the importance of large animal models, like the D50MD dog, which closely mirror the disease in humans, in contrast to the commonly used mouse models which show minimal clinical signs of the disease. The D50MD dog model has elevated levels of dystromy-RS in serum and significant fibrosis in skeletal muscles, like humans with DMD. They state that quantifying miRs in muscle of D50MD dogs compared to wild-type dogs would help better understand both dystromy-RS and fibromy-RS in the disease. The authors also note that suitable, stably expressed reference RNAs are necessary for accurate quantification of miRs using reverse transcription quantitative PCR, or RT-qPCR, to account for variations introduced during sample processing and the RT-qPCR procedure. They cite a lack of validated reference miRs in the literature, and emphasize that the appropriate reference miRs will likely vary depending on the species, age, and specific muscles analyzed. They mention that snRNA U6 is commonly used, but has variable expression across species and disease states, which can lead to unreliable results. They previously found that miR223 was stable in D50MD and WT-controlled dog serum samples, but emphasized the need to establish appropriate reference genes for each combination of species, disease, and tissue. The study was designed to identify stable reference miRs in the skeletal muscle of the D50MD dog model of DMD, across different ages and muscle groups. The dogs used in this study were from the D50MD colony at the Royal Veterinary College. Wild-type and D50MD dogs were housed together. The study followed arrived guidelines and was approved by the Royal Veterinary College Local Animal Welfare Ethical Review Body. A total of 8 male D50MD and 9 male wild-type dogs were included in the study, and followed from birth to 18 months of age, as part of a broader natural history study. The sample sizes were determined to give robust variance estimation for future studies. Muscle samples, specifically vastus lateralis, were collected by open biopsy at 3-month intervals. Additional muscle samples including cranial tibial, semimembranosus, lateral triceps, and diaphragm were collected post-mortem from the dogs euthanized at the end of the study. Muscle samples were snap-frozen and stored at -80 degrees Celsius. MicroRNA was extracted using a Chiagan mRNA kit. RNA concentration and purity were analyzed using a nanodrop 1000 spectrophotometer. MicroRNA was reverse transcribed using a Mercury LNA-RT kit. For the initial screening, cDNA from vastus lateralis samples of 4 wild-type and 4 D50MD 6-month-old dogs were analyzed using the Dog MeFinder Focus V2 panel. This panel contained primers for 87 miRs of interest, RNA spike-ins, and an interplate calibrator. Quantitative PCR was conducted on a light cycler. The resulting data were analyzed using GNorm, BestKeeper, and NormFinder algorithms to identify potential reference miRs. Based on the initial results, a subset of 8 potential reference miRs was selected for further study using individual qPCR assays. These assays were performed using cDNA from longitudinal vastus lateralis samples, as well as post-mortem muscles from 3 wild-type and 3 D50MD dogs. The selected reference miRs were CFA miR15A, HSA LET7B5P, HSA miR29B3P, U6 snRNA, HSA miR106B5P, HSA miR130A3P, CFA miR191, CFA miR125A, and HSA miR27B3P. Interplate calibration was achieved by using a single plate to quantify a subset of miRs and samples from each original plate. Following the selection of reference miRs, qPCR assays were performed using primers to miRs of interest, specifically CFA miR1, HSA miR133A3P, HSA miR206, and HSA miR214-3P. Statistical analysis included unpaired, two-tailed t-tests to compare results between wild-type and D50MD dogs, as well as linear mixed models to compare results between muscle groups and different ages. The results section details the findings of the study. Figure 1 illustrates the muscle samples used for RT-qPCR assays, showing the vastus lateralis, cranial tibial, semimembranosus, lateral triceps, and diaphragm muscles, the age of dogs at time of sampling, and dog IDs. Gray boxes on the figure represent samples that were not available for analysis. The initial MeFinder panel analysis identified 14 of the 87 candidate RNAs with very low or absent levels and were omitted from further analyses. The remaining 73 candidate reference genes were analyzed using GNorm, BestKeeper, and NormFinder. GNorm analysis, which assesses the expression stability of the candidates, revealed a wide range of stability values. Many candidates scored well within the commonly accepted threshold for suitability. The assessment of wild-type or D50MD samples alone lowered the M values globally, indicating considerable disease-associated differences. For instance, miR7 and miR106A were highly ranked in wild-type but scored poorly in D50MD, while miR26A and miR126 ranked highly in D50MD but poorly in wild-type. Members of the LET7 family including LET7C and LET7B scored well overall and in D50MD dogs. miR191 and miR125A also performed well across all 3 groups. In contrast, snRNA U6 scored poorly, ranking 51 out of 73 for all samples. Figure 3 depicts the GNorm M expression stability values for the MeFinder panel with results shown from least stable to most stable for RNA expression in the vastus lateralis muscle from 6-month-old dogs. Part A shows results for all samples grouped together, B shows wild-type samples alone, and C shows D50MD samples alone. Table 1 lists the top 20 potential reference RNAs by base plus GNorm analysis for all samples, wild-type samples, and D50MD samples highlighting RNAs common across all 3 groups. BestKeeper analysis also showed that miR191 and miR125A were highly ranked. However, only miR181A was among the top 20 in all groups. snRNA U6 performed poorly, ranking 34th, 67th, and 42nd for all samples, wild-type samples, and D50MD samples, respectively. Figure 4 depicts BestKeeper Pearson correlation coefficients for the MeFinder panel, showing results from least stable to most stable for RNA expression in the vastus lateralis muscle from 6-month-old dogs with A for all samples, B for wild-type samples, and C for D50MD samples. Table 2 lists the top 20 potential reference RNAs by best keeper analysis. NOR-finder analysis also showed high rankings for miR191 and miR125A and low rankings for snRNA U6. Figure 5 shows NOR-finder values for the miFinder panel, with A for all samples, B for wild-type samples, and C for D50MD samples. Table 3 lists the top 20 potential reference RNAs by NOR-finder analysis, highlighting RNAs common across all three groups. The initial miFinder panel results were used to identify 8 potential reference miRs based on consistent high rankings from the 3 algorithms. These miRs included miR130A, miR191, miR125A, LET7B, miR15A, miR27B, miR29B, and miR106B, while snRNA U6 was also included. Individual qPCR assays using the broader muscle sample collection were performed to identify the most suitable references from the shortlist. GNORM analysis using this wider sample collection showed greater agreement across subsets. miR125, LET7B, and miR191 consistently scored highly, while miR29B and snRNA U6 were the least stable. Figure 6 depicts the GNORM values for A, all samples grouped together, B, wild-type samples only, and C, D50MD samples only. Table 4 lists the GNORM results for the panel of 9 potential reference RNAs, also presenting data for a variety of sample groupings. Under best keeper analysis, miR29B and snRNA U6 ranked poorly across the board, with the exception of miR29B in wild-type 18-month samples. Figure 7 depicts best keeper Pearson correlation coefficients for A, all samples, B, wild-type samples, and C, D50MD samples. miR15A performed well, along with LET7B, miR125A, and miR191. Table 5 shows the best keeper results for the panel of 9 potential reference RNAs, including results for a variety of sample groupings. NOR-finder analysis revealed that LET7B, miR15A, and miR191 ranked in the top 5, with very high stability. Figure 8 shows the NOR-finder stability values for A, all samples, B, wild-type samples, and C, D50MD samples. Table 6 shows the NOR-finder stability values rankings grouped for a variety of conditions including genotype, dog ID, age, or muscle. The authors validated their top 4 reference miR candidates miR191, LET7B, miR125A, and miR15A by using them to normalize the lowest scoring miR29B, and they also compared it to using snRNA U6 for the same purpose. Raw expression of miR29B did not significantly differ between wild-type and D50MD samples. However, when normalized to either the geometric mean of the top 4 performing reference miRs, NF4, or to snRNA U6, miR29B expression was significantly lower in D50MD compared to wild-type samples. Furthermore, the authors found an age-associated increase in miR29B expression in both genotypes when using the NF4 normalization method, with young D50MD muscle having higher levels than healthy muscle, but subsequent increases with age being more modest in dystrophic tissue. These distinctions were not apparent when U6 was used as a reference. These findings are shown in Figure 9, including log relative quantity of miR29B expression A, raw B, normalized to NF4 or C, normalized to snRNA U6. D and E show results grouped by genotype, muscle, and age for NF4 normalized and snRNA U6 normalized miR29B, respectively. The raw, non-normalized snRNA U6 expression was modestly, but significantly higher in D50MD samples compared to wild-type samples. Post-hoc analyses revealed significant differences at 9 and 15 months of age, along with an age-related decline in snRNA U6 expression. These findings are shown in Figure 10. Following normalization of U6 to NF4, significant differences were still detected, both between genotypes and muscle group. Conversely, no such differences were seen in the geometric mean of the top 4 reference RNA candidates, NF4. When each of the top-performing reference miRs was examined individually, using NF4 for normalization, There was no significant effect of genotype on miR191 or LET7B. Conversely, miR125A and miR15A exhibited small increases and decreases, respectively in D50MD compared to wild-type dogs. The expression of dystromiRs miR1, miR133A, and miR206, and fibromyr miR214 were measured using NF4 for normalization. miR1 and miR133A were significantly lower in D50MD samples, while miR206 did not significantly differ between genotypes. Fibromyr miR214 was significantly elevated in D50MD samples. Figure 11 shows the log relative quantity of expression of A, miR1, B, miR133A, C, miR206 and D. miR214, normalized to NF4. Parts E through H show results grouped by genotype, muscle, and age for the respective miRs normalized to NF4. The discussion section of the article begins by reiterating that accurate quantification of target miRs requires the use of normalization factors. The goal of this study was to identify and validate reference miR candidates for a range of skeletal muscle groups from the D50MD canine model of DMD and wild-type control dogs of different ages. The authors used a two-tiered approach with a primary screen to identify strong candidates, followed by an in-depth secondary analysis using those strong candidates in a large number of samples. The primary screen used 6-month-old vastus lateralis samples from wild-type and D50MD dogs because this age has shown the onset of reduced muscle mass and resistance to eccentric exercise, along with a peak in serum biomarkers of muscle damage and inflammation. The quantification of 87 miRs in these samples produced a panel of potential reference miRs, based on stability scoring by the algorithms GNorm, BestKeeper, and NormFinder. The three algorithms share many common RNAs in their top 20, including miR130A, miR191, miR125A, miR125B, LET7B, LET7C, miR27B, and miR106B. They note the variation between genotypes, with miR191 scoring uniquely well across all algorithms. Members of the LET7 and miR27 families scored well for both genotypes, with LET7B, LET7C, and miR27B scoring as the most stable overall. They also note that miR30D scored well in D50MD and miR20A scored well in wild-type dogs, but were not consistent across genotypes and may merit further investigation as potential therapeutic biomarkers. The authors shortlisted miR130A, miR191, miR125A, LET7B, miR15A, miR27B, miR29B, and miR106B for further testing. Despite its poor initial performance, they also included snRNA U6 as a commonly used reference RNA for context. The shortlisted panel was then analyzed with the much larger collection of muscle samples. The vastus lateralis muscle was chosen for the longitudinal biopsies because it is easily accessible and also commonly biopsied in human DMD patients. Post-mortem muscles were selected to ensure representation of pelvic limb, thoracic limb, and respiratory muscles. Across the three reference gene assessment programs, the majority of the panel exhibited very high stability, with the exception of snRNA U6 and miR29B. miR191, miR125A, and LET7B scored exceptionally highly for stability, regardless of genotype, age, or muscle group. miR191 has been proposed as a universal reference miR due to its stability across various tissues. Several studies have shown dysregulation of miR191 in cancers. miR125A is also abnormally expressed in cancers, suggesting a role as a tumor suppressor. The LET7 family of miRs are highly conserved and play a role in cell differentiation, with lower expression of some members associated with poor prognosis in cancer. On the basis of these results, miR191, LET7B, miR125A, and miR15A were considered suitable candidates and used as a normalization factor, or NF4, for miR expression in D50MD and wild-type dog muscle, regardless of age or muscle type. In contrast, miR29B and snRNA U6 were the least stable. snRNA U6 is widely used as a reference miR, despite studies showing variable expression across disease states and cell types. They emphasized that miR223, which was a suitable reference miR in their previous study of D50MD and wild-type dog serum, scored poorly for stability in skeletal muscle, demonstrating tissue specificity. The study showed that snRNA U6 is not suitable for miR analysis in wild-type or D50MD skeletal muscle. The second lowest ranking RNA, miR29B, was shortlisted due to its strong best keeper stability scores in the primary screen, despite not performing as well under GE-norm or NormFinder analyses. miR29B has a role in skeletal muscle atrophy and antifibrotic roles. The authors showed that miR29B was not a suitable reference miR for D50MD or wild-type muscle and was significantly altered in D50MD muscle. The authors also normalized each of the 4 reference miRs individually to the collective factor, NF4, to assess their respective contributions. There was no significant effective genotype for either miR191 or LET7B. However, miR125A was increased slightly, while miR15A was decreased to a similar extent in D50MD dogs. The authors suggest that miR191 and LET7B could be used individually or in combination, and that multiple reference RNAs are recommended. The geometric mean of all 4 validated reference miRs was used as their normalization factor. The authors used NF4 to normalize expression of dystromiRs miR1-133A and -206, and fibromiR miR214, and found that miR1 and miR133A were lower in D50MD, while miR206 did not significantly differ. This is similar to results found in DMD patients but not in mouse models. They suggest that the lower dystromiR expression might be due to loss of muscle tissue via atrophy/fibrosis, while the elevated serum dystromiRs could be markers of muscle fiber integrity rather than a pathophysiological process within dystrophic fibers. The study showed that miR214, which has been linked to fibrosis, was elevated in all D50MD muscles at all time points. The authors suggest that, along with miR29B, this could be a valuable marker for muscle fibrosis in future preclinical trials. They conclude that the normalization factor derived from miR191, LET7B, miR125A, and miR15A is a highly stable and robust representation of total miR content, regardless of genotype, age, or muscle type. While multiple references are essential, they note that miR191 and LET7B would be suitable for investigators wishing to use a reduced panel. The authors revealed that snRNA U6 is unsuitable for normalization in this model. They note the value of the D50MD model, showing that the dystromiRs miR1 and miR133A are decreased in D50MD dogs, while miR206 is not, similar to DMD patients but not mouse models. The authors also show that miR29B is decreased and miR214 is increased in D50MD compared to wild-type dogs. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.